I would like to divide this presentation into five main parts. First, I'm going to look at DNA polymerase 1 discovery. Then I will move on its introduction, structure, function, and conclude the presentation with its mechanism. DNA polymerase 1 was discovered by Arthur Kornberg and his colleagues in 1956, just a few years after Watson and Crick proposed their hypothesis that there might be an enzyme that could replicate DNA. Arthur Kornberg did some wonderful experiments to test the hypothesis. He took DNA sample along with labeled DNTPs and added E. coli cell extract to this mixture. If the cell extract had any enzyme that could sorry, if the cell extract had any enzyme that could replicate the DNA, then the labeled DNTPs would get incorporated in the DNA, which can be easily detected. When he did the experiment, the hypothesis was found to be correct, which shows that there is an enzyme in the cell that adds DNTPs in the DNA. He further purified this enzyme by various chromatographic techniques and named this enzyme as DNA polymerase 1. In 1959, the Nobel Prize in Medicine was awarded to Arthur Kornberg. DNA polymerase 1 or Pol1 is an enzyme that participates in the process of DNA replication. It was the first known DNA polymerase 1. It was initially characterized in E. coli and is predominant in prokaryotes. In E. coli and many other bacteria, that gene that encodes Pol1 is known as Pol-A. It is an example of processive enzyme. It can sequentially catalyze multiple polymerization steps without releasing the single-stranded template. DNA polymerase 1 is a single polypeptide chain with 928 amino acids. It has three sites which provide three distinct catalytic activities. DNA is bound to PAM when the enzyme is active. It is catalyzed by magnesium ion. The finger domain functions to bind the NTPs with the template base. The thumb domain plays a potential role in the processivity, translocation, and positioning of the DNA. DNA polymerase 1 has three major activities. Number 1, 5' to 3' polymerization activity. Number 2, 5' to 3' exonuclease activity. Number 3, 3' to 5' proofreading or editing activity. DNA polymerase 1 is involved in the removing RNA primers in the processing of DNA after replication. 5' to 3' exonuclease activity means the polymerase is going to remove the nucleotides in 5' to 3' direction. This occurs when the polymerase identifies small single-stranded cuts on the DNA, which are also known as nicks. The 5' to 3' exonuclease activity and 5' to 3' polymerization activity occurs simultaneously, and this process is known as nick translation. DNA polymerase 1 also fills in the resulting gaps by synthesizing DNA beginning at the 3' end of the neighboring Okazaki fragment. For this process, polymerase requires a primer that can be extended in 5' to 3' direction. This process occurs in the presence of magnesium ion. The hydroxyl group at 3' and loses its hydrogen and form an oxyanion, and in presence of magnesium ions, the oxyanion reacts with the alpha phosphoryl group of the DNTPs and completes the reaction. Both DNA polymerase 1 and 3 have the ability to proofread their work by means of a 3' to 5' exonuclease activity. This activity is useful when the polymerase adds wrong nucleotides in the DNA. The 3' to 5' exonuclease activity halts the polymerase and removes the wrongly added nucleotides. This avoids mutations in the newly replicated DNA. Unlike DNA polymerase III, DNA polymerase I can use DNA as well as RNA strand as template to synthesize new DNA strand. This diagram depicting the role of DNA polymerase I during the process of DNA replication. Thank you for